Hello, everybody, and welcome to another instance of our journal club, Closer Look, an open journal club in biomechanics. And this is hosted bi-weekly by Emma Lejeune, Matthew Versi, Manuel Rausch, Johannes Backemeyer, and myself, Adrian Buganza-Topole. And today we have the honor of having Deva Chan, who is a colleague of mine at Purdue in biomedical engineering, who did her PhD actually at Purdue before starting. She did also a postdoctoral position in NIH, I believe, and then was at RPI before coming back to Purdue recently. I want to say 2019.
2020,
but
middle of the pandemic. It all blurs together.
In the middle of the pandemic, and yeah, and now she's my colleague, and she's gonna talk to us about functional MRI for mechanics. So very interested in learning about her work. And without further ado, it's Deva Chan.
Right. Thanks, Adrian, for the introduction. All right, so I was asked by the organizer to talk specifically about functional MRI can detect changes in intra-tissue strains in a full thickness and critical sized ovine cartilage defect model. But I don't think I can fully do it justice without giving you the framework in which it existed. So I'm actually going to start there. And I always like starting with this slide just because I think it's quite sobering for those of us that have knee joints and might be growing and aging. And a lot of what I do in my group, now that I have my independent faculty position, is kind of motivated by these increases in osteoarthritis specifically, but also arthritis in general, that we're going to see in the American population and worldwide as people get older and also as people get more overweight. And so as you can see here, when this study was done, probably in the 2010, 2015 range experimentally, and then published somewhere in this range, we had an estimated 50 to 60 million people in the US with osteoarthritis. And you can see that as we move into the future here, 2030 and 2040, that number gets close to 80 million. And if you work out just the simple math, that's about 1 1/4 of the total adult population is going to have some kind of doctor-diagnosed osteoarthritis or arthritis, right? And of those doctor-diagnosed arthritises, osteoarthritis accounts for over 16.5 billion in hospitalization costs, and that was over 5 years ago. So you can imagine how much more it's gone up now, especially as people are less prone in the last couple of years to see their primary care physicians and go in to get checked up. And if you look at just the chronic pain numbers, it's actually 1 in 3 people over the age of 65 experience chronic knee pain, right? And so we know that there's various comorbidities to osteoarthritis as well, including obesity, heart disease, and other inflammatory diseases. And so like I like to tell my lab, it's all linked. That's why we study all of it. But specifically what we look at in our group is the knee. And inside the knee, there's this delicate balance that is rather beautiful of all the biochemical and mechanical factors that are necessary to maintain a healthy joint. And so if something disrupts any of these mechanical or biochemical factors, you start it along this pathway towards an osteoarthritic joint, which is shown on the right here, right? Osteoarthritis is a disease that in more recent memory has now sort of progressed into this whole joint disease model. But in earlier times and decades ago, it was really thought of as a wear and tear disease, particularly on the articular cartilage. And so a lot of the focus in osteoarthritis research has really been focused on looking at articular cartilage and the degenerative changes that occur in those tissues as osteoarthritis progresses. So there's several risk factors that are currently known to increase your risk for osteoarthritis. Some of these I consider to be immutable, so things that we can't necessarily change about a patient, but we can at least be able to tell that patient that they're more predisposed to OA in the knee. And these include things like genetic factors as well as deformities of the joint that increase the stress in different parts of the cartilage in the knee or elsewhere. There are some that are very complex factors. So they include potentially things that the subject or the patient can't change, but also some things that may potentially be modifiable. And I group these as sort of these complex factors that are in between, like demographics, especially things like what you eat, populations that you might come from, as well as movement biomechanics. So anything that can alter the way you go about your daily kinematics. And then there's sort of squarely what I consider to be lifestyle factors that are associated with increased risk in osteoarthritis. And these are obviously obesity, existence of a prior injury, as well as overuse of the joint, right? And so for all of these things, these are actually, they tend more towards the biomechanics side of things, right? These are factors that have in some way altered the biomechanical behavior of tissues in the joint and lead to potentially degeneration down the line. So ultimately, all through my grad work and including now, one of the questions that really drove my research is what changes mark this early and progressive osteoarthritis, right? Like, what are these changes that we can look for in order to track the progression of the disease? And if we know what we're looking for, once we've detected it, is there a way that we can go back to either a healthy joint or at least stop that progression? And so it's worth knowing if, well, in our case, right, we're doing imaging as well. And so in order to really understand what we're doing the imaging for and what we're trying to look for in these joints, it's worth understanding what these early changes are in osteoarthritis. And we're interested in early osteoarthritis. So before I move on to the next page, we're interested in early osteoarthritis because currently there's no way to really stop osteoarthritis once we begin to detect signs of osteoarthritis. And so the earlier we can detect it, the higher chance we have of being able to slow the progression or stop the progression of disease. And that's because at the very end stage of the disease, you have high degrees of joint stiffness, high degrees of patient pain when it comes to mobility, and at the end of the line, when it comes to OA, what you end up is having patients that need to have their joints replaced, right? And so there's a host of other issues that occur, and nobody wants a major surgery. And so we want to be able to treat it at an early stage, and that means we want to be able to detect it at an early stage. And so this early disease is marked by several compositional and structural factors, right? And those are the ones that are more generally studied. And these are, well reviewed elsewhere, but I'm going to go over some of the ones that will be relevant for us today. Early on, in the cartilage, there are development of surface fibrillations. In terms of the cartilage matrix, you see increased water content as well as swelling of the cartilage matrix. And usually this swelling isn't quite enough to be able to be detected as a change in overall cartilage thickness, right? But some studies have shown just early enough that you might, once you get to a disrupted enough extracellular matrix in the cartilage that you can actually detect some of this swelling. And I'll talk about that in a bit. You're also going to see changes in the bone, including increased bone porosity as well as remodeling. And that's accompanied by changes in the phenotype of the chondrocytes as well as of the cells in the bone itself. A little bit later, you begin to see things that are a little bit more detectable non-invasively, like a decrease in proteoglycan content and a disruption in the collagen network that makes up the articular cartilage. You can also see changes in the cortical plate thickness of the bone that's directly underlying the cartilage. And then you also see other changes in the subchondral bone, including some of the changes in the ultrastructure and the architecture of the bone. And then before you get to mid and late OA, some of the changes that you really do begin to detect more macro scale are the fissuring and fragmentation of the matrix. So you actually have delamination or fibrillation of the articular cartilage. And this is where you begin to see thinning of the articular cartilage, as well as the formation of osteophytes, which are bone spurs on the periphery of the cartilage. And also the formation of bone cysts and other changes that are existing in the bone. And so with this progression of changes, and I've summarized them all here, with this progression of changes, we want to be able to detect these things early, right? And so as I talked about, with very, very early OA, a lot of these techniques that are able to detect these changes aren't currently available in terms of non-invasive assessment, right? No patient is going to allow you to do an AFM on their existing cartilage in order to determine whether or not the surface is fibrillated. And the same thing happens with any of the histology and other benchtop assays that tell us whether or not there's changes in cell phenotype or changes in water content or swelling of the matrix. And so it's not until you get to this sort of second stage of early OA that some of these things begin to be detectable outside of the body, or at least at a tissue scale. And so things like MRI as well as CT will be able to detect structural changes and biochemical changes in the proteoglycan content and the collagen network. And then with CT, you begin to see some of these bony changes, like the cortical plate thickness increasing, and then also changes in the architecture in the subchondral bone mass. And then finally, before you get to that very last stage of early OA, if my arrow, things went too fast there. You actually do begin to see these things on radiographs as well as with standard MRI and not just specialized sequences, right? So on MRI, you begin to see thinning of the cartilage and changes in signal intensity. And on radiographs, you begin to be able to recognize that you have changes in the bone, including osteophyte formation, bone cysts, and then also loss of joint space. And the one thing that I do want to point everybody out to is that, at least in osteoarthritis, all of these changes usually occur before any loss of function is noticed by a patient and before any pain actually occurs in that patient most of the time, right? And so that adds to the challenge of early detection and intervention in osteoarthritis because the patients aren't even coming in at this stage. And so that's sort of always something that I want to make sure that is noted, even if we're working on these imaging techniques to enable detection of early OA, that we're also limited by how the patients behave and when they come into the clinic to see their clinician about joint pain or joint discomfort. And so all the things that I talked about also manifest as altered biomechanics, right? If we have this increase in water content, which was that first phase, or the loss of proteoglycan and disruption of the collagen matrix, these are all going to change the way that the cartilage responds to a particular load, right? And so obviously, I'm not going to go through all these bullet points, but as biomechanists, we know that changes to the extracellular matrix are going to change the overall material properties of that tissue, right? And so at the cartilage level, we know that these things change, and it's similar in other tissues as well. We know that along with these changes in biomechanics and biomechanical behavior, you end up getting changes in mechanobiology. And that's why the other portion of my lab now looks at mechanobiology as well, because it's those mechanobiological changes that further precipitate the progression of OA, right? Thus incentivizing us to stop it early on. And so it can do that in a various number of ways, from the cell scale all the way to changes in joint biomechanics and gait. And then, of course, eventually all of these changes change the pain and the function of the joint overall in terms of stability and mobility, right? So all along the progression of OA, these changes that are associated with the progressive degeneration of the joint can also manifest as altered biomechanics. And so thus the incentive for us to be able to measure these changes in biomechanics in concert with all these other biochemical and cell phenotypical changes. And so that kind of leads sort of to the end of my introduction here in terms of what we do as a lab. And a lot of that is, guides how our lab approaches studying joint health and disease. And so of course we have, we're engineering still, so we're kind of centered on the biomechanics. But from some, a lot of my research background, we also bring in medical imaging and some matrix biology as well, right? So how does the extracellular matrix change and how do the cells respond to those changes as well as elicit those changes? And so today we're mostly going to focus on displacement encoded MRI, which sort of sits at this interface between biomechanics and medical imaging. Although given some of the questions that you guys had prepped for me, we're going to start talking about how this intersects with matrix biology and some of the other aspects as well. And so we'll talk about imaging. The imaging technique that my lab focuses mostly on, at least at the joint scale for cartilage imaging, is magnetic resonance imaging. And it's a very good tool for healthy cartilage as well as early OA cartilage, because you have a high degree of signal that comes from articular cartilage due to its hydration, as well as the inability for other techniques like CT to really detect non-calcified tissues. And so let's talk a little bit about how MRI is currently used in the field for cartilage and cartilage repair. And so if you take conventional MRI, so standard MRI, what you think of is changes in structure and overall damage that you can see. One thing that you can see as well is changes in water content. So when you have bone marrow lesions like you have in this image here, you are able to see that in a fat suppressed image on the right as increased edema with that bone marrow lesion after a particular injury, right? With conventional MRI, you can also look at changes in cartilage thickness and volume, and also whether or not you have lesions in the cartilage. And that's true for any tissue that will show up on MRI and give you a signal on MRI. And usually in the clinic, the imaging of cartilage and the joint is usually paired up with semi-quantitative grading protocols that clinicians use to overall determine what the state of the morphology of the joint are. But these are usually restricted to signal intensity as well as apparent damage to the tissues in the joint. If we move beyond what's typically used in the clinic, there are also several other quantitative MRI techniques that are out there. And a lot of these correspond to various tissue properties. So prior studies have shown a lot of work, or at least correlations to extracellular matrix content. So you can imagine that if water content goes up and down, it will change the signal. And the same thing is true depending on what quantitative MRI technique you use in terms of changes in proteoglycan and changes in collagen content, as well as collagen ultrastructure. And so far, a lot of the consensus from the field is that these quantitative MRI techniques are actually much more sensitive to early OA progression than radiographs, right? So X-rays, or even the structural findings that we see from conventional MRI, right? And so you can see changes in some of these quantitative MRI measures earlier in the progression of OA than you can some of the changes in thickness and volume. In addition, there's a technique called sodium MRI that instead of imaging protons, which are heavily abundant in water and fat, what you're actually directly imaging is the quantity of sodium in a particular tissue. And for most tissues, this doesn't come up very much, right? Because you have a very low sodium content. But fortunately for articular cartilage, there's a high glycosaminoglycan content, and GAGs, glycosaminoglycans, are highly negatively charged and therefore have a strong fixed charge density. And of course, sodium is highly positively charged and tends to equilibrate into areas that are highly negatively charged. So you get a strong sodium signal just from the presence of more sodium in the interstitial fluid, as well as in the tissue of cartilage. And of course, that changes as you get degeneration in the tissue as well. So as you lose glycosaminoglycan content, you lose your sodium MRI signal because of the change in fixed charge density. And then finally, going into the field that I'm in is ways to image joint tissue biomechanics using imaging, right? And admittedly, it's sort of underdeveloped, although it's not impossible. And so I'm going to talk about some of that work today, but I think it's important to recognize that it needs more work and more development, because you're able to assess both healthy and diseased tissue function and not just the content. And it allows you to be able to non-invasively, right? So in cartilage as well as in other tissues, it allows you to be able to non-invasively estimate what the tissue properties might be. And it's also a really good indication of how well a tissue repair might be integrated, right? And so none of the techniques that are here above, they're all correlated to the tissue content, but none of them particularly look at things like interface or interface biomechanics or even integration of a repair to the native tissue. And I'll talk about that a little bit in terms of our future work. So the one that for some reason got hidden in my slideshow was this one, right? And so early on in the development of this work in measuring biomechanics of cartilage using MRI, we came upon this technique called displacement encoded MRI. And it was initially developed for those of you that do more cardiovascular mechanics, you're probably well aware of it, but it was initially developed in cardiac tissues because cardiac tissues don't require an external actuator in order to deform inside the body in living human, right? And so it paired very well with imaging that enabled you to be able to measure the strains in the ventricle and the atria in vivo. And so that was the challenge that we set upon ourselves. Are we able to measure some of these same mechanics using the same imaging techniques, but now doing so in other tissues that don't have self-actuation? And so very briefly, this is how displacement encoded MRI works, right? We essentially time our external loading, which occurs across one particular load cycle, and we time that with our imaging. And within every single load cycle, which is also a load imaging cycle, during what we would consider within that cycle, our reference configuration, we use the radio frequency signal on the MRI system in order to excite the tissues of interest. And what that means is that part of that excitation includes turning on and off the magnetic gradients that allow us to encode the position in X and Y within that frame of every single pixel on that image, right? And so all of these things occur in our reference configuration. You have protons that are now within these tissues that have a phase that is directly proportional to the position that it's in this reference configuration within our load cycle time. And then when we apply our loads, which are going to be the same across every single loading cycle, that is when we acquire the data, right? And so we have our RF acquisition. We are able to basically apply the opposite gradient here. So we call it position decoding rather than position encoding. And so we're effectively taking away the phase difference that we applied earlier, again, based on the position that a particular proton is in X and Y within that plane. So for protons that don't change in position, when we encode and then we decode, you have an overall zero change in the phase information that proton possesses. However, when you have a proton that changes in position, right, you have either a net gain or a net loss in terms of the phase. And that gain or loss in phase correlates directly to the net change in displacement in the X direction if we turn the X gradient on, and in the Y direction if we turn the Y gradient on. And that's essentially your 5-second primer there of how displacement encoded imaging works. And so when we acquire that data, right, we get our MRI data out. MRI data is typically in magnitude information as well as phase information. And we take this phase information that was embedded and we extract what the displacement in the X direction as well as the displacement in the Y direction was based on the information that we encoded and decoded within each cycle above. From there, we have our discrete displacement maps from which we can calculate our deformation tensors and then calculate our strain fields. And so it's this entire process that allows us to be able to measure the displacement of a particular pixel, right? So it's on average all the protons within that pixel. And we're not necessarily constrained by the spatial resolution of that because the change in position is actually connected to the phase information and not where that pixel has physically relocated to. And so we did this in a host of different ones, but I think what's the most interesting one is always, doing it in living humans. Grad students that might be tuned in might recognize this picture as asking fellow grad students to volunteer as guinea pigs for science. And so what we did here was designing a MRI compatible pneumatic system that enabled us to load approximately 1/2 body weight to the foot. The picture is great. It's a little easier on the schematic to sort of see what's going on. And so what we have is, the subject lying down on a angled back support to prevent motion of the subject in and out of the MRI as they're being loaded, right? Rather than just pushing the person up the gantry and down, pulling them back down the gantry. The back support enables us to have some sort of resistance to that load. We have thigh and shin constraints in order to try to help fix the knee flexion angle. And of course, none of this is perfect because we're doing this in people that have to still also be comfortable. So we can't necessarily fix rigid body motion all that much. And then finally, we have the cyclic load being applied via a soft boot at the very end, aligned ideally to the tibial midline. And that load propagates through from the foot to the ankle, to the knee, all the way to the hip, where hopefully you're not getting any more motion because the subject is no longer moving because of that back support. And so we validated the system outside of the MRI because there's a lot of measures that we can't take except outside of the MRI because it requires load cells and displacement sensors that have magnetic components that we can't put into the MR. But essentially what we're able to do is for the 5-second duration cycle is measure the overall displacement of the foot with respect to the back. And then in that loading plateau, we're able to measure the displacement and also the spread. So the blue is the mean and the lower and upper 95th percentile. So are indicative of the spread of the data between cycles. And so we determined based on this how many cycles we had to put a particular subject through before we reach some kind of quasi-steady state response to the load, right? And we wanted to make sure that we hit that quasi-steady state before we began loading and imaging at the same time. And that's because we want the images to be consistent and in the steady state portion of the cyclic loading. And then we also took a look at the standard deviation as a function of the cycle time in order to really refine where across the load cycle we actually wanted to be able to image the data so that we had the least amount of variation. Because unfortunately with MRI, you have to take more than one load cycle and therefore more than one image cycle in order to fully acquire a single image. And so now that we have sort of a good idea of what generally happens, so I encourage, if you want to look at the in vivo data, that's in this paper here, but that gives you an idea, hopefully, of what generally happens during a displacement encoded MRI experiment that is timed with externally applied loading. And we can sort of shift to the experimental setup that was in the paper that we described. And that one, the goal of that paper was really to sort of take a step back and look at whether or not this technique would enable us to be able to detect differences in strain maps when you have a defect versus no defect, right? And so we went back to the drawing table here in terms of designing a different pneumatic cylinder that allows us to better control the loads that we apply to the joint. Obviously, when we're doing this ex vivo, we don't have to contend with a lot of the other soft tissues that might be in the way because we could just remove them from the sheep tissue post-mortem. And so here you can see that we have a pneumatic cylinder. It's a double acting cylinder, which allows us to compress the joint as well as retract for our unloaded portion of the cyclic imaging and loading cycle. And we potted both the tibial as well as the femoral end of the sheep stifle within the constraints of our MRI loading system. And we did all this on a 7T, so it's a higher field magnet, which allows us to get much better resolution in the sheep versus what we can get in a 3T system with the human. So the higher the magnet strength, the better resolution we're able to get. So we really wanted to be able to do this as high strength of magnet as was possible, as a proof that we can measure strains in a defective cartilage. And so the loading was very much similar. We still did the 5-second loading cycles, and we still loaded it up to about 1/2 of the body weight of the sheep. And you'll see here that, and I'm pulling these images now from the paper for those of you that have the paper up with you as well, so that we can sort of talk along with that. But we're doing this loading cycle, and we're timing that with the events of encoding. So here is where we're doing the encoding. This is where the decoding occurs. And notice that it's being decoded during the load plateau. And then it's also being decoded during the time that we're acquiring the data using a sequence called TrueFisp. And so everything is timed in terms of the load as well as the imaging in this cycle. And we took a look at the 4 different specimens. And so obviously this is what the specimen might look like afterwards. After this entire test, right? We took the specimen, and this was actually just a picture that we took for the purposes of the paper. And this is not actually a specimen that we used for testing, because you'll see that it's disarticulated, and we did not disarticulate any of the samples for this. But it does get the point across in terms of what tissues we're looking at in the intact version compared to the defect. And we used essentially a drill to create a 8mm diameter osteochondral defect. And so our study was essentially to do MRI, standard MRI of the intact joint, load it until it hit the quasi-steady state load deformation response, and then perform dual MRI, which is displacements under applied loading with MRI, in order to measure the displacements in the cartilage. And after that first study was complete in the intact joint, we would open up the joint, create this defect, and then repeat the experiment shortly afterwards. And so what we found, well, I guess this is sort of one example of what we found, right, was that, we had our intact joint here on the left, and then our defect on the right. We can see that we did not fill the defect with anything. And so because this is all happening outside of the body, it doesn't fill up with any fluid or other tissues that you would get if you were to perform a defect in a living sheep. And so when you see this black space, it's essentially air in this case. And so we looked at all of the cartilage, but we also keyed in on the cartilage contact area, and that was different actually for every single sheep, depending on how that geometry lines up. But we always did try to put the defect in the middle of the medial condyle for all the sheep. And so these displacement maps are what come out after we do dual MRI. So our displacements under applied loading with MRI. And, obviously in the intact joint, what you'll see here is, just from looking at the very smooth colors, or not the smooth colors, but the very similar colors, is that a lot of our displacements are dominated by rigid body motion of the tibia as well as the femur, right? And so even with our very tightly constrained geometries from here, sorry, my slides keep moving around whenever I do this, right? Even with our very tightly constrained geometries, there is still some concomitant motion when it comes to looking at the overall displacement of these tissues, which is something that we saw in the human studies as well. And then we did it the same thing in the defect. Fortunately, we saw that a lot of the rigid body motion is still constrained by the joint capsule. And so it's very consistent between the intact and the defect. And so we're able to focus in on the strains that actually occur within the cartilage in both the intact and the defect cartilage. And so we did a couple of things before we got to the strains, right? When we see the raw displacement, so this is one of the example of the raw displacement from the tibial cartilage, you'll see that we still have a bit of noise, right? And as anybody who's done strain maps based on displacement might know, you have to be able to eliminate some of that noise and have a smoother displacement map before you can calculate the strain, or else you're going to get very spurious strain numbers, right? Because you have the influence of noise rather than actually having this one pixel, for example, displacing in one direction while the rest of the cartilage displaces in the other direction, right? And so points like this are very much noise rather than the true displacement. And so the first thing we have to do is figure out a way to smooth it. And so for this study, we started, our previous work had looked at just Gaussian smoothing. And so it's the same no matter which direction you look at. But especially as we found in the human study and then now in the sheep study, one of the important things is that if you're smoothing with a Gaussian filter all across it, you're losing a lot of through thickness changes because of the way you're smoothing, right? You're smoothing the same way in every single direction, but your tissue itself has a very different aspect ratio, right? And the way that we implemented the Gaussian was that it would stop smoothing as soon as it hit a border, because obviously the displacement in the bone is not going to be averaged in with the displacement of the cartilage. And so for this study, we tried a newer technique, or a different technique rather, and that used locally weighted linear regression instead, in order to smooth out some of the spurious displacement points. And so with this lowest smoothing, it preserves a little bit better some of this through thickness gradient that you might expect in terms of the strains, right? And we expect this because we don't anticipate that anything that's near the relatively rigid bony contact to be changing as much as the displacement of the articular surface of the cartilage where we are compressing, right? And so we expect that you're going to have this gradient in displacement just because you have a rigid surface on one side and you're getting contact from, in this case, the meniscus on the other side. And so that gradient is lost when we do Gaussian smoothing, but it is preserved at least when we do locally weighted linear regression. And you can sort of see that gradient here as well in the raw displacements, but of course it is corrupted by these points where you have higher noise at the borders. And so with this newer smoothing method, we were able to then calculate these in-plane strain maps. And so this is just another representative specimen with the intact as well as the defect. And you can appreciate that with the defect, we see changes in the strain profile in both the tibia as well as the femoral cartilage. And we see some of that as well when we calculate the principal strains, in-plane principal strains, as well as the in-plane maximum shear strain. So this is one of our representative subjects. And so when we look at them across the 4 different samples that we used here, we can sort of plot out what happens to these max and min principal strains in plane, as well as the max shear strain for both the femoral as well as the tibial region of interest. And of course, we can do the same thing, not just for the entire region of interest, but also for only the cartilage contact zone. And despite the changes in the strain field that we saw when we grouped all of these together, because of the high variability, which I'll talk about in a bit, we really only saw significant differences in the femoral contact region, right outside of where we created the defect, right? And so there, with the intact versus defect, we saw significantly higher strains in the femoral cartilage, for both the first principal strain as well as the maximum shear strain. And they trended, of course, in the same direction, since those are related parameters. And so you can look at the paper for this one, but basically the main takeaway from this table really, from these 2 tables really, was that when we looked at these average principal shear strains across the entire region, we saw overall increases with the defect, but it was highly variable between the 4 different sheep we had. And so when we sort of looked across as a population, some of the strength of these changes didn't really pop out at us as much, right? And again, that's probably because of some of the limitations of the study and that technique. And so, as I already alluded to, the location and the placement of the defect were very much dependent on the geometry of the sheep. We had centered it on the meniscus, but that was no guarantee that that was where the cartilage contact was occurring in the intact joint, because of the position of the meniscus as well as other tissues. And so across the different sheep, the placement of the defect with respect to where the cartilage and cartilage meniscus contact regions differed, right? And that led to a difference in terms of where these strain concentrations might occur and how great these strain concentrations might have been. Of course, a lot of that came from the inter-sample variability that we saw between the different sheep, as well as potentially any differences in material properties of the tissues themselves between these different sheep. As I also mentioned, we are looking mostly at in-plane displacements and in-plane strains, and that is primarily constrained by the total imaging time that's possible for the current technique that we were using at the time, right? And so it became infeasible. In order to get 3D volumetric, like through the entire volume of the joint, you would have to do multiple slices, and that would effectively, for every single set of slices, you would have an additional set of scans that you would have to do. And that became very infeasible in terms of total imaging time, especially as we, with the intact joint, we aren't able to inject any protease inhibitors into the synovium. And then, finally, these were obviously done post-mortem. And so what we're able to compare with intact versus defect is really just what would happen after the initial creation of the defect. So we're unable in this particular case to assess what the repair or the regeneration process might look like after an injury has occurred, and then after the attempts to repair it. And that kind of leads to our future directions as well, right? And so our future directions kind of link up with a little bit of what the previous limitations of the studies are. A lot of these differences in specimens are inter-subject variability differences, right? And so if we're able to integrate these with subject-specific computational biomechanics models, instead, it enables us to be able to use the imaging data as inputs for both the construction of the model as well as for the various boundary conditions that might exist with that particular load setup as well as that particular geometry, and then be able to calculate or better describe what the tissue biomechanics might be if we were able to normalize these tissues to each other a little bit better. Of course, we're still working on the imaging aspects as well. So trying to accelerate the imaging and make it more feasible to do it in a shorter imaging time using techniques like TrueHarp as well as compressed sensing. And this is absolutely necessary because if we want to do longitudinal studies, which is what we're going to be able to need to do in repair and regeneration processes, we need to be very careful about the overall imaging time that we have, especially if we're starting to do these in live animal or potentially live human studies. And in order to make that transition, of course, we're going to always start at high field where we get better resolution and better signal. And then we're going to start with an in vitro repair model first to test out whether or not our technique works and is able to track things like the maturation of repair tissue and its integration with the surrounding tissues. And then what that enables us to do is also begin to integrate other quantitative MRI techniques that will provide us with complementary information beyond just the biomechanics information. And so finally, I did want to just end with sort of pulling us back out in terms of the perspective, right? My lab looks at a lot of different research directions, and I sort of just talked about one of them because we look at biomechanics as well as a lot of other topics of interest that sort of overlap at that interface of biomechanics, imaging, and matrix biology. And I could happy to talk about that more as well. And then as usual, my acknowledgement slides is true for any project that I do. But the work, well, of the people on here, the work that was done that I presented on for this study was done in collaboration with my former PhD advisor, Corey New, who's now at University of Colorado. And so that's it for me. I can bring it back to, I'll plug my lab. So I'll bring it back to this one as I answer questions.
That was awesome. Awesome. Super interesting stuff. A little bit frightening as well, to be honest, that we're all gonna get arthritis. So
someday we're all gonna get it.
But this was amazing. I'll open it up for questions from the audience.
Yeah. So I see one picture, one in the chat. How much time delay is there between calibration and actual MRI measurement? I am not quite sure at what slide this appeared on. So if Matthias could
it was
interject.
a slide where you were showing the cyclic loading for the human study.
This one?
Because you cannot really know the one before.
This one?
Where
I think after, yeah, that one.
Yeah, that one.
Okay. So calibration for that particular subject?
Yeah, so before, like, as you said, like you do the measurements first to define your cyclic count and like the displacement, and then.
So what we generally do for all our studies is we take a representative sample from the person and we find out how many cycles we need to get to steady state for that subject. And we take the pool of our subjects and we take mean plus 2 standard deviations. And that's the point where we actually start to image, right? Because there's no way that even if we do this right before, we would have to be able to transport the subject to the MRI, and everything changes once that happens. And so for all of our studies, we always do a whole set of calibrations beforehand to figure out what that n plus 2 times standard deviation is. And so we are pretty confident at that point that, you know, if we were to do 96 point whatever samples as subjects for that many of them, we would have hit quasi steady state. And so that is a limitation, right? Because we don't know what it is for every single exact person. But fortunately, once they're in the MRI, one thing that we're also able to do is take standard scans during loading as well as during the unloaded portion of the cyclic cycle. And we'll do this about every 50 cycles as we approach what the steady state's supposed to be. And we'll actually take, you know, load cycle 100 minus the image from load cycle 50, and we'll start to see how the image inside the joint converges as well. So for us, that's a second check to make sure that we've hit this sort of steady state response, quasi steady state response, before we actually do any imaging.
I have a quick follow-up to that before we move to the next question. So how long in total for a given person or for the sheep does it take if you have to do, is it like 100 cycles times 5 seconds or much more than that?
So for the human, we did want to limit it, right? Because we couldn't have them in the MRI forever. And then whenever there's a reflexive muscle motion, you would also change the quasi-steady state, right? So that's why the standard deviation for this is much higher than what we would have seen in the sheep. And so if I remember correctly, I actually don't remember the exact number. It's in the low hundreds of cycles, so we're not keeping them in for too long. And so I think if I remember correctly, we spent about maybe 10 minutes doing the essentially preconditioning before we did the imaging, and then imaging itself took about another 15 minutes to get a full slice. And then for the sheep, again, I don't remember the exact details. I could probably bring up the paper instead, but but it was pretty similar. We were able to do it for much longer for the sheep, but I don't think, in both cases, we verified with imaging of that specific specimen before we actually started.
I see. So, but rough order of magnitude around like 20, 30 minutes to.
Yeah, roughly like the scan will take about 20 to 30 minutes. And then, you know, if you can get to quasi steady state faster, you can begin imaging faster, right? And so you can, you know, depending on what your load parameters are, you can sort of accelerate that. But you can, you know, you also run into other issues with balancing image parameters and loading parameters there too.
Thank you. We have a
still pretty decent. I would be willing to spend that time to know. Right? If you want to know how.
Right. We would be willing. I think our standard subjects might not be willing to know.
That's true.
Yeah. So Vahidullah asks this: endurance running increase your risk of osteoarthritis? Not my area of expertise, but from what I remember of the literature I have read, it depends on how you define endurance running, like how many miles and how often it is. You know, recreational running actually reduces your risk of osteoarthritis because exercise is good to prevent OA, and it also helps to maintain cartilage health. But at a certain point, you sort of hit this diminishing returns in terms of running, and you're actually creating more stress, especially when you start going into marathon and higher, I would say. But you'll have to check the literature on that. That's sort of just my general impression of having read a lot of the exercise plus cartilage preservation literature.
See, Mariana turned her video on. So Mariana, do you have a question?
That's the universal cue, isn't it? Hey, Deva, great talk. Really exciting to see this work coming together. I have two questions, and they're kind of the big bird's eye view hard ones. I was curious what your, if you have an opinion on whether the subchondral bone changes precede the cartilage changes or the other way around.
Yeah.
Do we know yet? And how do we use this to kind of figure that out, or are they happening together? I guess that's question one.
So my opinion is that they happen together.
Okay.
And that's, still to be determined, I guess. So I would like to repeat some of so we did a DMM study with micro CT in mice, where we both imaged the bone as well as the cartilage, right? So with CT, you have micro CT, at least you have a much better resolution for the bone, so you can start looking at those trabecular changes as well as subchondral bone plate changes, as well as using contrast agents for your cartilage. So that's the kind of study that you can do on both. And there we found that they sort of started around the same time point, right? And so of course it's limited by DMM because you're opening up the joint and causing a host of systemic inflammation or local inflammatory events that might affect bone more strongly than it might affect the cartilage as well. But at least from what I know so far, they happen at the same time.
Did you look at markers of bone turnover in that as well?
No, we didn't look at them. And so that was done mostly in conjunction with a sort of a bone histomorphometry group. So they were very much more concerned with changes in the actual like bone structure and cartilage structure. But when we do those experiments with our non-invasive ACL rupture, hopefully we'll be able to do some of the more biology side of things.
Yeah, that would be super cool.
Yeah.
And
I'll tell my students that to sort of see if I can push them along. If it's, multiple people think it's super cool, you should do this study earlier.
Yeah. Well, I think it's still heavily debated, right?
It is.
What's really what precedes what, and the mechanisms. So
yeah, and then all the crosstalk, and there's a lot there.
Exactly. My second question was kind of my ears pricked up a little excitedly when you mentioned at the beginning of your talk about your desire to go beyond measures of content and really look at function of tissues.
Yeah.
And can you tell us what you think the function of the tissue, or what is the functional marker that you're trying to go for? Is it like a strain function, not a strain function, but like, are you looking for certain strain thresholds that the tissue would stay under? Is it a biomechanical function that you're talking about, or
so for
or something else?
Right. So for this context, I guess for MRI, obviously if we're getting the strains, I think having some degree of a threshold where you might start seeing damage would be good. None of the studies we've done so far have sort of exceeded that yet, right? Even the defect study, they haven't exceeded what people have described in vitro models as generating cartilage damage, right? And so I think, while that's good to keep in mind, I don't think necessarily we'll be ever generating the kinds of strains that would cause damage in a cyclic loading situation, right? Because you can't damage the tissue and then keep imaging it, expecting the same mechanical response, right? You'll never hit that quasi-steady state because you're just continually damaging the tissue. And so that's where it gets tricky, right? So I think, part of it is going to be starting to look at ways that we can incorporate biomechanics modeling into calculating what the material parameters are, rather than just what the mechanical response to loading is. But being able to use some of this non-invasively measured biomechanical behavior in order to inform that model. And so then that might be a threshold of shear modulus or, aggregate modulus that we can then use to say that, well, there's damage, because modulus is decreasing to this range instead. And so I think at the tissue scale, it's still a little questionable for us, right? What the marker actually needs to be, because given the constraints of the imaging, we haven't quite gotten there to be able to measure strain as a threshold.
And how do you think that relates to pressure then, in terms of load distribution at the surface?
Yeah, I mean,
is that another marker?
Like I think so, right? Because if you have an increased contact area, you can extrapolate things from an increased contact area between two opposing surfaces with cartilage as well, right?
Yeah.
And so that's part of why we want to move beyond just a single slice, because we're missing a lot of out of plane and volumetric information here. But unfortunately, with imaging, we're sort of constrained until we can figure out how we can either use improvements in image processing or improvements in image acquisition to go faster, so that we can get more information, or at least figure out in which planes or what angles we need to get that information, so that we can optimize for image time as well as information gathered.
Yeah, it's super cool. And ultimately, when you go to dynamic conditions, that's when you'll be able to really start doing some interesting things, looking at dynamic motion, right?
Yeah. And so I think ultimately we would probably need to partner up with people that do gait biomechanics as well, right? Because you would do this at a particular known flexion angle. We can match that up with what happens kinematically, and then be able to really build that model that can, not just better describe what's happening throughout the gait cycle or throughout a particular motion like squatting, but be able to potentially predict where you might see damage 5-10 years down the line too.
Yeah, very cool. Thanks.
Awesome. All right, I'm going to end up the official journal club, but for people that want to stick around, if Deva has some time, I don't know. But for now, since we are right at the hour, I'm going to say that this is a conclusion of today's journal club. Thank you everyone for joining. Thank you, Deva, for that excellent talk and discussion afterwards. I think this was amazing. So yeah.
Yeah, thank you guys for hosting, and thank you everyone for attending.